Okay. Hello, everyone. Thank you very much for the invitation. I'm very grateful. It's a great pleasure to talk to such a broad audience. So my talk today is about the population landscape. Let me start with giving you an overview of what we are doing at the Biotechnology Center in Khalifa University, headed by Dr. Habiba Al-Safar, and where I am the bioinformatics team leader. So we've just published the first whole genomes from the UAE in Nature Scientific Reports. Secondly, we also study the genetic diversity and the population of the United Arab Emirates using still an old-fashioned technique called like SNP chips. And I'll try to convince you that it is actually a good idea to combine both strategies. We also use these SNP chips for having a look at the population structure analysis. And further, now that more genomes are coming in, we are aiming for developing the reference genome. And we also are developing techniques to do variant prioritization with respect to our local population. So I'm particularly honing in on the three topics framed here. Let me start with two initial concerns when we do our work. So we now have, we all know that the local or the GCC and genomics landscape is pretty understudied. We only have 0.08% in public databases. Another concern is that maybe we all talk about whole genome sequencing or next generation sequencing being democratized and being more and more available, but a lot of effort has been shifted towards the analysis part, as you can see here in the yellow bar or the orange bar, the lower part. The third column is actually showing you the amount of effort being put into the actual data analysis, which is now taking more and more of the whole process. Okay, our motivation is, as I mentioned already, is to develop a strategy to elucidate our understudied local genetic landscape in both depth and breadth. And this obviously is a good foundation for finding better biomarkers, gene panels towards personalized medicine. Regarding depth, I think we have a good approach by using very comprehensive SNP chips, namely the Illumina Omni exome SNP chips with 4.5 million exonic variants. And this also gives a lot of breadth. Namely, we have managed to get data from 1,000 Emiratis, so this is quite a significant number in order to be getting a good impression of the local landscape. Here are the bioinformatics steps that we are doing in order to analyze our data. So we start with, or I mainly focus here on the SNP chip or genotype array data that we have from the Omni exome 5 chips, which we then submit to Blink for the usual quality control. Then we further do admixture and principal component analysis of our data, and also we have a tool that actually combines the two. I will show you in a second. Then further, we look at the allele frequencies, which we then also annotate with what's known worldwide. So for this, we look at the NOMAD database, but also at the Greater Middle East study that has been performed some time ago in order to get an idea of how the allele frequencies, are they much lower, are they much higher than worldwide? And this is a relatively comprehensive approach for millions of loci. So then we developed something called the Z-score, which gives you not just the raw allele frequency or the absolute allele frequency, but also sets things into perspective. I'll talk in the next slide on this. Finally, we collect our data in an SQL database that is facilitating also annotation. The typical ones are SNPF for having an idea on what is the variant doing. Is it exonic, intronic? Is it synonymous or non-synonymous, and so on. And once we have that database, we can derive a bunch of data, query, we can do sophisticated querying on that, and which we then use to build up a Variome browser. A little bit on the z-score, it's a bit technical here, so it tells you basically how far from the mean is my data point, in our case our allele frequency, and it divides it by the standard deviation, so you can basically say how much is our allele frequency away from the mean in terms of standard deviation units. And this puts our allele frequencies in a global perspective. So the other allele frequencies, as I mentioned, are taken from the NOMAD database. So the rationale here is to maybe have a look at so-called known biomarkers that have low allele frequency worldwide and high allele frequency here, which would yield a high z-score. They are unlikely to be causative. We often see that gene panels or genetic testing comes back negative, and that is something we might have been might be able to predict with our tool. Conversely, we also see that rare variants, those that have a low z-score and comparatively low allele frequency in the Emirati population, they might be less permissive. I don't want to say too much, but they might be less permissive in our local population, and we would like, in a study of millions of variants, we would give them a higher priority. Okay, so here's, let me start with the results. Here's a general picture, or the general landscape, as you've probably seen with the typical principal component analysis of our local population. I hope it's visible, the circles that have actually a circumference, these are our local samples contextualized with populations from the Human Genome Diversity Project. And what you also see is that our samples are shown with pie charts. And these pie charts show you the actual admixture that you normally see in those typical Q matrix plots. And what is remarkable in this little inlet here, you see, you see in the inlet, you see that we have a relatively broad spread of our local population along the internal axes. These internal axes are usually spanned by the African and the Asian component. This is actually interesting to also compare to, for example, other studies done in the GCC, but also nearby in, for example, Armenia. The third picture is from a study in Armenia. You often see that the local population ends up in the apex of the two axes of the Asian and African apex. But what I would like to say, the main take-home message we have from our study is that we have actually a wide spread and also a genetic continuity in comparison to the other studies. So I wouldn't talk so much about clusters, but rather argue that we have a very admixed population and relatively a relative genetic continuity there. So not really clear clusters, which we believe could be the result from undersampling in other studies. Okay, in total, we have from our SNP chips 2.3 million exonic loci after quality control, And we also use them to inform our whole genome sequencing. We want to, once we have the big picture of the entire landscape, we can then cherry pick, or not cherry pick, but we can choose very representative parts. Also, we identified 46,481 variant loci that are significantly different than in the rest of the world. And we express that in terms of our z-score. So if our z-score is greater than 4 or less than -4, which also corresponds to a very small p-value, then we think we can have a deeper look into them. And we believe that they are unique biomarkers, probably for medical purposes, but definitely for anthropological and forensic studies. So what else do we find in them? So these 46,000 variants, we have split them up by a SNP-F effect. So they're not, you could argue that they are, most of them not really doing anything, possibly, but we do actually see some of them that do change the coding sequence of the proteins, and we also observe stops gained and start codon gained. So there are quite a significant number of those. We can also look at the spread of those UAE-specific variants. So as you would see here, we list them per chromosome. They do actually spread as you would expect. You see a little peak in chromosome 6, which is not so unexpected, given that there are some hyperpolymorphic regions like the HLA and the MHC. Yeah, so but you see a pretty widespread view of our variants over the genome. It's also interesting to look at the entire distribution. So here you have a z-score histogram, which basically tells you so how many special variants do you have. So right now we were looking at z-score 4. It's a bit arbitrary. We can also look at a broader circle. Once we have prioritized our variants, we can also look and enlarge the circle of potential interesting variants by looking at z-score 3 or -3, which brings the numbers from, for example, 45,220 high allele frequencies, supposed to be high allele frequencies, to 71,000. And on the low allele frequency side, we're going from 1,261 to 3,312. If we extend it even further to a z-score of 2, we would end up with hundreds of thousands, for example, 128,000 allele frequencies that we have particularly higher here in the country. What I would like to show you also here is a tool that we developed based on those allele frequencies. We have here the so-called Variome browser, where we also where we list up, we map the allele frequency or the loci. We study them by metabolic pathway. So for each metabolic pathway, you can see what are the UAE-specific variants here. So here you see some examples that show you the allele frequencies in the UAE and the rest of the world, and you see many of them. So here we have an exonic one that is very high in the UAE, and you can study the SNP-F effect, and you can also go to GNOMAD. Okay, so going a bit further, we have the 1,000 exomes now by imputing, doing imputation. The imputation can heavily benefit from local data. We have as a reference panel, not just the 1,000 Genomes project, but now we have a lot of local data. We have a particular focus on exonic regions because we have a lot of exonic reference data from the region, from the GCC. And this is actually an ideal case for pan-Arabic collaboration. Okay, an idea for future data deluge is to go towards big data technologies. We would like to mention that we have strong ties to the people who developed OpenCGA, which is also used for the Genomics England and 100,000 genomes in the UK. which actually uses true big data technologies rather than the flat files and SQL databases that you've previously have seen. So to round up, so this is for, if you expect like 64,000 genomes, so then this is the kind of technology we would like to recommend. To summarize, we have now our population study landscape completed, and we would, okay, I think time's up. I'd like to acknowledge my team and Dr. Habiba Al-Safar, and I'm ready to take questions. Thank you very much.